AS Level Biology 9700 Paper 2 Variant 2 February March 2026, and in this video we will discuss the question number 3, and question number 3 is raffinose is a sugar found in many plant species, and you can see the figure 3.1 which shows the structure of one molecule of raffinose. So this is the long chain of raffinose, and question is raffinose is formed by the reaction of three different monosaccharides, one of which is glucose. So on figure 3.1, draw around the part of the molecule that forms a glucose when the glycosidic bond in raffinose are hydrolyzed. So here you can see that this raffinose molecule is made up of three monosaccharides. The upper one, this one is galactose. And the lower one is the fructose. And this is the glucose. So these are the three monosaccharides which make up this raffinose molecule. Now, part 2 of this question is state the type of the glucose molecule that is formed when the glycosidic bonds of raffinose are hydrolyzed. So we have to write the type of the glucose molecule which formed is that is alpha glucose. And how can you say that this is the alpha glucose? Go to the figure 3.1, and you know in alpha glucose the OH group is in the downward direction. So looking at the middle ring, the glycosidic bond from carbon 1 projects downward to connect with fructose. So here you can see when it joins with the fructose, so it means the OH was in the downward direction. So we can say it indicates that the original hydroxyl group on carbon was in alpha position, that is pointing in the downward direction. So this is the alpha glucose. And next question is raffinose produces the same results as sucrose when it is tested with Benedict solution. Complete table 3.1 to show some features of the sugar, glucose, maltose, and raffinose. Maltose is a disaccharide. So number of glycosidic bonds in one molecule as the disaccharide molecules are formed when two monosaccharides join together. So it means when a glucose is a monosaccharide, so there will be no glycosidic bond formation into it because it is a single molecule. So we will write 0 over here, and in the it is reducing sugar. Maltose is disaccharide. It means two molecules join together to make one maltose molecule. So here I would like to say that it is, there will be the formation of one glycosidic bond, and it is maltose is also a reducing sugar. Raffinose is the molecule which is made up of three monosaccharide molecules. So there should be the two glycosidic bonds by which they are joined with one another. So this is non-reducing sugar. Moving towards the next question. Some bacteria in the human digestive system synthesize an enzyme known as alpha GAL, alpha gal, so which breaks down raffinose. The rate of breakdown of raffinose by alpha gal at different concentrations of raffinose is shown in figure 3.2. So you can see this graph. Now determine the Michael-Menten constant for alpha gal using the data in figure 3.2. So here you can see that V maximum, they have shown us the V maximum, which is at 6. This point is Vmax. And half of the Vmax should be between 4 and 2, that is at 3. Half Vmax, which is at 3. So now if we want to find out the Michael-Menten constant, then we have to go towards this in this way, in the downward direction. Let me do it again. It will be in this way, and here, from here, we will move in the downward direction. So it will be equal to here, and which is 0.02. 0.02 on this graph. And it can be accepted from 0.02 onwards, 0.04. It will be accepted. This range will be accepted according to your marking scheme. Now, because it will be different in different sizes of the pages. Now, next question is, one of the molecule formed from the breakdown of raffinose by bacteria is shown in figure 3.3. So you can see this COOH is there, COOH, which is called as a carboxylic acid group. So state the type of molecule shown in figure 3.3. So according to this, I would like to say this is a fatty acid or saturated fatty acid, and COOH, presence of COOH group shows that it is carboxylic acid. So we are moving towards the last part of this question. The molecule shown in figure 3.3 can be used to make a triglyceride. Describe how this molecule can become part of the triglyceride molecule. Uh, you know that in triglyceride molecule, condensation reaction takes place in which the fatty acids reacts with the glycerol molecule, glycerol head, and the fatty acids will be the tail. So it undergoes the condensation reaction, which involves the removal of water molecules, and bond formation takes place. So the hydroxyl group of the glycerol reacts with the carboxyl group of the fatty acid to form a covalent which is ester bond. So three fatty acids join a single glycerol molecule to form a complete triglycerol molecule. I would like to light over there. By a condensation reaction, which involves removal of water molecule and formation of ester bond takes place. So three fatty acids join single glycerol molecule to form a complete triglyceride. So it was all about question number 3. Please like, share, and subscribe my channel to watch more videos on this paper. Thanks for watching.